Hello everyone. Today we will discuss blood typing, and importance of ABO groups in blood transfusion. Importance of ABO blood groups in blood transmission. Next one, RH blood types. RH blood types. First one, blood typing. First one, blood typing. First one, blood typing. So the blood typing means, so the in phenotypes ABO phenotypes. So blood groups in ABO phenotypes. So the blood groups ABO phenotypes. ABO phenotypes. Phenotype individuals mixing blood sample mixing with anti-serum mixing with anti-serum. So the blood sample mixing with anti-serum. Blood sample mixing with anti-serum. So in the anti-serum contain antigen A and antigen B is presenting. So the anti-serum contain antigen A and antigen B. So the serum contain. So the blood mixing with serum. So the serum contain anti-A and anti-B antibodies. They contain serum contain two antibodies. What is the anti-A antibodies? Anti-A antibodies and anti-B antibodies. So the blood sample mixing with anti-serum. Blood sample mixing with anti-serum. So the blood typing time. So blood sample mixing with anti-serum. The anti-serum contain two types of the antibodies. So one type of the antibody is the anti-A antibodies. Second one anti-B antibodies. For example, taking a slide. So these are the one slide. So in this slide, taken a blood sample. 2 drops of the blood samples. So these are the A, these are the B. So in this blood sample, so they are adding with the anti-serum. The blood sample adding with the anti-serum. For example, so these are the adding with anti-serum. So in this adding with anti-serum, only the formation of clumping. So clotting is formation. So this clotting only formation in the A sample. So mixing with only anti-A. So these are the blood group A type. Next step. So they are the sample, another person blood group. So they are the sample A, sample B. So adding with the anti-serum A and anti-serum B. So these are clumping only B type. In the B sample. So the person blood group is B type. Next another person blood group. Slide. So the samples are the 2 drops of the samples. So they are the sample A, they are the sample B. So they are adding with the anti-serum A antiserum B. So they are clumping in the both the A and B. They are clumping with the A and B. That person's blood group is type is AB type. AB type. Next one. Slide. Sample A and sample B. A and B. So they are adding with the antiserum. Adding with antiserum. So in these two samples, the two samples, there is no clumping, there is no clotting formation. So this blood group is called O type. This type is called as O type blood group. So they are the blood typing. So these are the blood typing. So first step, so the blood sample mixing with antiserum. The antiserum contains two types of the antibodies. One type of the antibodies, anti-A antibodies, second type anti-B antibodies. So they are the only anti-A sample, they clump, they are anti-A sample, so they are the formation of coagulation, clumping is formation. That type blood group is called as A type blood group. Next one, so they are the mixing with only formation of clump in the B type, so their blood group is called as B type. So the clumping is formation A and B, clumping is formation A sample and B sample, so the blood group is called as AB type. Next one, so there is no formation of clumping, so they are called as O type blood group. So they are the blood typing. Next one, importance of ABO transmission, importance of ABO groups in blood transmission. Next, second one. Next one, importance of ABO groups in blood transmission. Importance of ABO blood groups in blood transmission. First one, so the consideration. So in the blood group transmission, so they will be transfusing the blood. The type and types of the antigens of the donor. Type and types of the type or types of antigens of the donors. Next one, type or types of antibodies. Antibodies of the receiver. So they are these two is considered. So first of all, they are considered from the blood transmission. So the donor antigens are the considered. Importance of the blood transmission. Antigens are the donor and antibodies of the receiver. Antigens of the donor and antibodies of the receiver. These two is the importance of the blood transmission. So they are the importance of, they are the consider of the blood transmission. Blood transmission. Next one, so the antibodies of donor and antigens of the receiver. So these two is less importance. Less importance of blood transmission. So in the blood transmission time, major importance of the donor antigens, receiver antibodies are the major importance of the blood transmission. So the donor antibodies and receiver antigens are the less importance of blood transmission. So the blood transmission time, only antigens of the donor and antibodies of the receivers are the considered. So they are the importance of the blood transmission. So the antibodies of the donor and antigens of the receivers are the less importance of blood transmission. Next one, so the blood group, O blood group. O blood group is called universal donor. Universal donor. So the blood group, O blood group, the blood group name O blood group is called as universal donor. O blood group is the universal donor because of, so the O blood group antigens are absent. So the O blood group antigens are the absent. So the AB blood group, AB blood groups. AB blood group is called universal receiver. Universal receiver. Universal receiver. So the AB blood group persons are called as universal receiver. Universal receiver because of, so in this AB blood group persons there is no antibodies. There is no antibodies. See the table once. Previous class we will see the table. So they are the blood type. Antigens on RBC. Antibodies of plasma. Antibodies of plasma. First blood group A type. First blood group A blood group. So in this A blood group persons the antigens presenting on the RBC, antigen A. Antigen A is present. The antibodies, anti-B antibodies. Anti-B antibodies. They contain anti-B antibodies. Next one, B blood group type persons, their antigen B is present. Antigen B is present. So the antibodies are the anti-A antibodies present. Anti-A antibodies. Anti-A antibodies is present. Next one, AB blood group. Next one, AB blood type. So in this AB blood type, antigen A and antigen B is present. Antigen A and antigen B is present. Antigen A and antigen B present. But so the antibodies are the absent. Antibodies are the absent. Next one, O blood group. Next one, O blood group. So these are the antigens are the absent. Antigens are the absent. Next one, anti-A and anti-B antibodies is present. Antibodies is present. So the anti-A and anti-B antibodies is present. So in this O blood group is called as universal donor. O blood group is called as universal donor because of antigens are absent. Antigens are absent. So the O blood group is called as universal donor. O blood group is called as universal donor because of, so the antigens are the absent. Next one, so the AB blood group is called as universal receiver. AB blood group is called as universal receiver. So the because of universal receiver, because of, so these are the antibodies are absent. Antibodies are absent in the AB blood group. AB blood group antibodies are absent. So the blood group is called as universal receiver. So the O blood group is called as universal donor because of antigens are absent. So they are the blood transmission importance. So the O blood group donate the all the type of the persons. A blood group donate from the A blood group, B blood group, AB blood group and O blood group. And AB blood group receiving from the universal receiver. AB is the universal receiver because of, so they are receiving from the A blood group, B blood group, AB blood group and O blood group. So they are the importance of blood transmission. Next one of the important word. So the AB blood group is a universal receiver, but so the AB positive, only AB positive, either positive and negative blood groups. Only AB positive. AB positive is universal receiver. Universal receiver. OB positive is AB positive is universal receiver. Next one, O negative. O negative, the universal donor. O negative, universal donor. So in this word, so the O blood group is universal donor, AB blood group is universal receiver. In deep study, so the AB positive only, AB positive is universal receiver, O negative only universal donor. So they are the importance of ABO groups in blood transmission. Next one, RH blood types. 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 So the RH blood types, so the scientist, so the Carl Landsteiner, scientist, Carl Landsteiner and Alexander S. Wiener. So two scientists, one is the Carl Landsteiner and Alexander S. Wiener. In these two scientists, so discovered by the RH antigens. So discovered by the RH antigens. So they discovered the RH antigens. So these RH antigens are first time discovered in the, so the RH antigens. So the RH antigens is also called as RH factors. So these RH antigens first time identify the rhesus monkey. The name rhesus. First time identify the rhesus monkeys. The first time identify the, so in this rhesus monkey. First time identify the rhesus monkey RBC. So the first time, so the RH factor identify the rhesus monkey red blood cells. In the red blood cell membrane, plasma membrane of the red blood cell, they contain RH factors. So rhesus monkey. So this antigen is called as D antigen. The RH antigen is called as D antigen. The D antigen. So the after after identify the rhesus monkey, the human beings are the identify the RH antigen. So the after first time they identify the rhesus monkeys red blood cells. After, so the human beings also, human being blood contain a RH factors. Human being blood contain a RH factors. So these are identified the Carl Landsteiner and Alexander S. Wiener. So these two scientists identify the RH factors the first time in the rhesus monkey red blood cells. So these blood groups, the RH factor is present in the RBC. RH factor is present. That blood group is called as RH positive or RH positive. That blood group is called as RH positive. Next one, in the RBC, RH factor is absent. RH factor is absent. That blood group is called as RH negative. RH negative or RH negative. RH negative. So the blood groups, so they identify the A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. So these are the positive and negative based on the presence of RH factor. That person's blood group is called as RH positive. Next one, that person's blood group, so the absent of the RH factor, that person's blood group is called as RH negative. RH positive and RH negative. So they are the RH blood types. RH blood types. Next class we will discuss so deeply positive and negative blood groups in the human beings.